Today, this brain pondering podcast is one that kind of historically overturned some dogma in a field, and that field is telomeres and telomerase biology. And my guest is Professor Gabriele Saretzki, Gabby, and she's recently retired. She was a professor at Newcastle University in the UK, and she started out working on cancer cells, I think, and normal proliferative cells, which essentially the telomere telomerase field focused on. And but more recently, she got interested in the potential roles of telomerase in neurons and in the brain, and that's going to be the main focus of this. So, Gabby, welcome.
Hi, Mark. Nice to see you.
And so you're in Germany now. You're from Germany,
Yes,
and
I am.
can you talk about like were either of your parents in science or medicine?
No, 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 no. I think I was the first of my family indeed to go to university. No, my dad was a joiner and my mother was a tailor. So
Wow.
pure, hands-on
So that
professions.
so they worked hard so that you could
Yeah,
go
yeah,
look for it.
exactly. Yeah.
Yeah, yeah. And then so you went to the Soviet Union for your undergraduate training?
No, that was 5 years essentially.
Oh, 5 years.
Yeah,
And
I was in a school which had where I learned Russian and English, so Russian from the third grade, and so that's why they sent me there. And yeah, it was 5 years, so that was essentially a bachelor and a master's together.
that was in molecular
St.
biology?
Peter's. Yeah, no, yeah, I mean, yeah, molecular biology. Essentially, it was a very broad training in biology, and yeah, St. Petersburg is a wonderful city, so I really enjoyed that.
Okay, and then of course at that time I was still, East and West of Germany, and there was the wall. So you went back across the wall to Berlin, is that right? From there?
No, I mean, I was at the eastern side, so I didn't have to cross the wall.
I,
So the Soviet Union
okay.
is at the
No,
eastern
no,
part.
no. Yeah, I'm mixed up. I'm mixed up. Yeah. So you would, but you went to Berlin then, Humboldt
No,
University.
I was born in Berlin, and yeah, I went back to the Humboldt University to the Department of Genetics, where I did my PhD then.
And so you learned a lot of methods there, mainly cell culture, or
No, no, I worked on yeast. So initially, I was at the Academy of Science of Berlin, and there I had to work on ribosomes in mitochondria. So that was the first time I encountered mitochondria. But then it was kind of in, I don't even know, in turnips, which were already laying there in the cold room for a while. And it turned out that there are no ribosomes in those mitochondria. So I changed tech and I went to work in yeast, essentially. And that was in more or less in molecular biology, as you said. But at those times in the 80s, the methods were when you wanted to isolate a plasmid, it took you a whole week with cesium chloride gradients. Do you remember?
yeah,
So that's not like you go to the fridge and take out the kit and then an hour it's all done.
yeah. And then you what? Then you went to Munich. Is that right?
No, I went to the charity, to the pathology. That's essentially the building where Rudolf Virchow worked. And there I tried to develop some methods to investigate viruses which were committed to HIV and other
Hmm.
such diseases. So it was quite applied, but I had a director and also a colleague, Thomas von Splinitzky, you might have heard of them, who were very interested in aging research. And at those times, aging research meant mainly measuring collagen in the tails of rats. And so, and and when telomeres came along, so that was really an exciting time because we essentially were there from the very beginning. So in the beginning, you had only one paper each week. And now it's thousands and thousands, you know. So yeah, that was exciting. So that was when we started with cell culture, still very difficult conditions, although the wall had come down by then. And yeah, so because before that was very difficult to get all these chemicals, you know, to do proper molecular biology. So yeah, but from then on, so yeah, I was interested in cancer research as well as in aging research. So, and with telomeres and telomerase being important in both of those fields, yeah. So that was quite okay.
And can you give some like basic information on what telomeres are and what telomerase is?
Sure. So the ends of eukaryotic chromosomes, they need a protection just as your shoelaces so that they don't unravel. And telomeres are tandemly repeated sequences in at least in humans and mammals, TTA, GGG. And so they can be 12, 10 kilobases, at least in newborns. And they also have some protective proteins. But since they shorten in every cell division due to the end replication problem, meaning the DNA polymerase is not able to replicate both strands at the same time. So at one strand, at the lagging strand, is always the RNA primer, which is used to synthesize the Okazaki fragments. It falls off. So that's why the telomeres would shorten every time a cells divide. And that's where telomerase comes into play, which is a very interesting and very exciting enzyme, which is able to counteract the shortening, which happens during DNA replication. So quite after that, in late S phase, telomerase, which is a very ancient enzyme consisting of the third protein unit and an inherent RNA, the TERC, so the RNA component. So, and being this, it's a reverse transcriptase using its own RNA moiety, which has a complementary template to the telomere sequence, and that's why it can synthesize telomeres de novo, at least in tissues where it's active. And that's the interesting part. It's not always active. It's mainly active during embryonal development in humans, but in other animals like rodents, it can be active throughout life. So it's quite exciting, but also sometimes complex and confusing. If people talk about telomeres and telomerase, you always have to be aware of the context, where it is active, where it's not active, and consequently what happens to it.
And right, and what's the link with aging? That was kind of an interesting development. What happens with telomere length and telomerase during aging?
Yeah, so I mean, first of all, most human somatic tissues after embryonic development shut down telomerase, probably due to epigenetic methylation of the promoter and so on. Only some human tissues like B and T lymphocytes and endothelial cells, of course, still tissue stem cells, they are able to upregulate telomerase. So in most somatic human tissues, the telomere shortening, which I tried to briefly describe, will happen. So that means telomeres shorten down. And as you know, in the 60s, Leonard Heflik, he found in the lab that human somatic cells, they have a limited cell lifespan. And later on, I think Carl Harley and colleagues, they found that it might be the telomeres would shorten down to critically short lengths, like around 5 kilobase, starting from the 10, 12, or 15 at the big start of life. And that means then the telomere is not longer kept by the proteins and loop structure. So it opens and it will be recognized as a DNA damage. And that's where the DNA damage response comes into play. And if the cell is normal and all tumor suppressors are functioning, P21, P53, P16, and so on, then the cell will stop. And that's what we call cellular senescence.
And can you describe and give a little more on senescence? So you have a dividing cell. The telomeres keep getting shorter every time the cells divide, and then some point can't divide anymore, and it becomes senescent. What does that mean, senescent?
So first of all, senescent is primarily being defined as a irreversible growth arrest. But that, of course, as we now know, only applies to dividing cells. However, there are other characteristics like changes in the nuclear chromatin, and most of all, it is connected to the senescence-associated secretory phenotype, meaning that there are a lot of different proteins and inflammatory, pro-inflammatory, and sometimes anti-inflammatory proteins are synthesized and secreted into the environment. And that means if you have in the tissue, a senescent cell can conduct a bystander effect. So it can also kind of infect other cells in order to also become senescent. And that's what essentially happens during aging. So in that respect, aging and senescence are connected. And
and then,
do you want
and so also, if you have DNA damage, it doesn't matter so much where it is in the genome, but in this case, the telomeres, the telomerase activity is down. It can't keep or
telomerase is not active in the first time.
There's this mechanism, these tumor suppressor gene, P53s, like the classic
exactly,
example.
It's the main one,
And it will, so when these telomeres get very short, the cells can either become senescent and start secreting these factors, inflammatory factors, or it can
go into
undergo
apoptosis
and be removed.
Absolutely,
So what happens then in cancer? I mean, these tumors are for example, mutations in P53 can lead to cancer.
So that's something which I think Jerry Shea was very is very well known for. So he defined these different pathways of mortality and immortality. So as I said, if P53 is active and telomeres are short and DNA damage occurs, senescence, you can also view it like a kind of anti-tumor mechanism. However, if now there are mutations in those tumor suppressor genes, P16 or also P53, which is probably the most important one, then these signals to stop dividing are overridden. So telomeres shorten further down, and then a lot of genomic instability can happen, but you also need at the same time oncogene activation or the inactivation of other tumor suppressors. And then cells can undergo crisis. A lot of cells will die by apoptosis or other means, but then there will be some, and that's now where the main culprit telomerase comes into play. Telomerase might be activated and might, although the telomeres are very short and they might even stay very short, but it's able to cap them by maintaining and stabilizing them and preventing these DNA damage responses. And so the whole thing can end up with cancer. And there was this very important study from Bill Hahn published in 1999, who showed it's very different whether you activate telomerase first in a cell which has active and functioning tumor suppressors. There you just maintain telomeres and the cells have an extended lifespan. However, if first genomic instability occurs, which already goes onto the path to tumorigenesis, and then telomerase comes, then it stabilizes the telomeres, but still within the chromosomes, all these genomic instabilities have already occurred, and they will promote tumorigenesis and later on maybe even metastasis and so. So these are, Jerry J called this M1 and M2 stages of immortality or mortality, from which side you view it.
I know Jerry Shea. I spent 2 years down in Texas, a master's degree, and he was down there in Dallas. Right.
He still is,
but then what, I got, I was as you know, when we're graduate students or postdocs or all the time, we're always, every week we look at Science, Nature, or
exactly. Yeah.
if you're a neuroscientist, you look at, well, now it's Neuron, Nature Notes. But anyway, so, I was doing this work trying to understand why neurons die in age-related neurodegenerative disorders, Alzheimer's, Parkinson's, etc. And so the telomerase story was kind of interesting to me because all the work was done in these proliferating cells where the telomeres shorten over time. But
absolutely, yeah.
But neurons, they're in your brain your whole life.
Yeah.
Right. And so exactly. I was kind of looking, as a neuroscientist studying these diseases, I want to keep the cells alive.
Sure.
Not dying.
Yeah.
But then I also got thinking that, okay, these cells aren't dividing, presumably their telomeres aren't shortening and, their death isn't caused by telomere shortening. And so I recruited a number of postdocs, and one was Wolfram Klapper from Germany, where you're at. He was at Kiel, I think, then, and he's actually an MD. And so he came to the lab, and my lab there's methods you can kind of quantify telomerase activity. There's something called a TRAP assay, telomere repeat amplification protocol.
Yeah.
So you use PCR, and then PCR.
Yeah, sure. I did this many times. Yeah.
Yeah.
With a gel and with a laser, and
so he did the simple thing. He looked at telomerase activity in during brain development.
Yeah.
And this is in I think it was rats then. Rats. It wasn't humans. And that's another interesting story. It turns out the rat and mouse telomeres are longer than humans.
Absolutely, yeah.
But anyway, the bottom line is he found telomerase activity was very high in neural progenitor cells, and then as they differentiate,
it decreases.
It decreased. But so that's telomerase activity.
Yeah.
The reverse transcriptase. But then he did Western blots, immunoblots, to look at protein levels for TERT, the telomerase protein. And there was still TERT in these post-mitotic neurons.
Yeah.
That's when we got interested in.
Sure.
And so back then there wasn't RNA interference yet. And so
that was 2002 published, I remember.
Yeah,
so you were probably the first describing that there's still TERT, but no telomerase activity, right?
Yeah. And then we used antisense. So essentially we reduced the amount of TERT, the telomerase protein, in these neurons, and found that made them vulnerable to a variety of stressors. So that was kind of that. And we didn't really pursue it too much more. So then at some point you entered the scene in terms of neuroscience, right?
Sure. Yeah, but that was much later. So I think we started like 2012. Esther Priel published this first paper on ALS. And at the same time we were working on it, and I thought, shit, she was the first publishing on the brain, so on brain tissue and on a neurodegenerative disease, and she's still very active doing this. So I think the two of us are those
that's
labs.
Another interesting story. So, we had proposed this based on our cell culture work, because we found that if we knock down telomerase, neurons are more vulnerable to being damaged by amyloid beta peptide. And
absolutely,
so on. But and so I got contacted by a PhD student with her, Era Zaitan.
Yeah, sure.
And so he did a postdoc in my lab on.
Oh, did he?
Yeah, something completely different, but.
Okay.
Yeah. So there's that connection. So can you talk about? So you really provided like another dogma breaking thing that. And we'll come back to this, but it turns out that so you and I did this work, and it kind of got buried in a way.
Yeah, I mean, I didn't.
But then more recently, some big shots in the field of,
Like
in a
Ron
general
DePinho.
sense, kind of confirmed and extended your findings and my findings.
Absolutely, yeah, sure, yeah.
So can you talk about, there's a lot of interesting things you did.
Thank you.
TERT and mitochondria.
Oh yeah, I mean, essentially, that was not even me. So it was Janine Santos from the States somewhere who found this mitochondrial targeting sequence. So because, I mean, you know, if people would have found TERT and mitochondria or telomerase activity, as we thought as well, if you don't purify the mitochondria properly, they always said it's an artifact. But she really showed that it is a biological process, and most importantly, it developed only later in the evolution of the telomerase, because I think it's not there in yeast or in plants. So it's something a telomerase acquired mainly on the path to mammalian cells. And that's very interesting, because that's one of the many non-canonical or non-telomeric function. I think there are that many gene expression and whatever complex it can be in with mTOR and whatever. So it's really a lot going on. So yeah, and based on this, we also looked into TERT protein in mitochondria, and at this time we also notified that there's no TERT, so no telomerase activity. And yeah, and we took it on from there. And after we published that in 2008, it also showed, most importantly, how it's shuttling. And there's also another German colleague of mine, you might probably heard of her, Judith Hendeler. She works in Düsseldorf, so she was in Frankfurt and Main at this time. And she also had very similar observations that TERT, and I think she was the one who described the TERT in order to leave the nucleus, and she showed it in endothelial cells, so where naturally you also have telomerase activity during senescence and during oxidative stress, that the TERT protein gets phosphorylated, or at position 778, don't nail me on the amino acid, by Src kinase, and that's why it can also leave the nucleus, and then the mitochondrial targeting sequence from Janine comes into play. So there's really, as always, a lot of different scientists playing a role in that. And then I had another colleague, a Japanese colleague, who worked with us in Newcastle University, and she said, yeah, so what if you look at telomerase in vivo, or TERT in vivo in brain, or for example in brain? I don't know how we got to brain, but at this time we also had some neuropathologists starting, Johannes Adams, so we had kind of a good environment to do this. And so we looked under conditions of dietary restriction, and we treated mice, so TERT knockout and TERT knockout and TERT wild type cells, also with rapamycin. And I have then had a master student, and she found that TERT also leaves the nucleus, although not so complete as, for example, under oxidative stress with H2O2, or if you culture cells under hyperoxia. But under rapamycin treatment, at least we used MCF7 breast cancer cells for that, just to visualize TERT better. Like 15% of TERT leaves the nucleus, and apparently might localize to mitochondria. And yeah, we really found that this decreases the oxidative stress in those brains from these mice. And yeah, and we moved on from there.
Okay, so you mentioned both dietary restriction, calorie restriction, or something we worked on a lot, intermittent fasting. And so those manipulations extend lifespan in rats and mice, actually by a lot, if it started when they're young. So this is like the classic anti-aging intervention. And I should point out the converse is true. If you overeat,
yeah,
it
sure.
shortens lifespan.
Yes, absolutely.
And then you also mentioned rapamycin, and
yeah.
we had recently had a podcast with David Rubenstein. He's at Cambridge, and he's done a lot of work
on autophagy, right?
Yeah, on autophagy. And okay, so this mTOR pathway, it's a mechanism whereby cells can take up a lot of amino acids and synthesize a lot of proteins. So when the mTOR pathway is active, cells are typically in a growing state.
Absolutely.
They're producing a lot of proteins. And then when you calorie restrict cells, animals and cells, then the mTOR pathway goes down, overall protein synthesis goes down. The cells kind of go into a stress resistance, conserve resources mode.
Yes.
And this rapamycin is very interesting. It has an interesting history. It was discovered in bacteria sample from Easter Island.
Yes.
There was this expedition from, it was actually multinational that went there. And
I read the book from Jared Diamond.
There's actually some controversy about this because there's these indigenous natives there. And there's this question, are they interfering with their normal? But anyway, and then a drug company, developed this rapamycin. It's used in the clinic now for immunosuppression with organ transplant.
Exactly, yes. Yeah.
But
that's right.
So I was at the National Institute on Aging for 20 years, and there when they started this, they had a 3-center program to screen interventions, mainly chemicals, to see if they extend lifespan.
Yeah, sure.
And the reason they had 3 centers, then you got the replication issue. And so they screened multiple chemical, antioxidants that squelch free radicals. They didn't extend, vitamin E.
No.
But the one thing that did extend lifespan was rapamycin.
Absolutely, yeah.
Not nearly as much as dietary restriction, but still it did. So you kind of found this link then between the mTOR pathway, telomerase, and mitochondria and oxidative stress. So what do you think? What do you think's going on there? The mechanism.
Oh, it's very difficult to say. So I mentioned the study with Satomi and also a talented Polish student, a PhD student, Rafał Czapewski, had this time. So we also found that under the conditions of rapamycin, the TOR pathway decreases, so which goes again along with this. And we did different kinds of experiments also, intervening with the TERT exclusion, with bosotinib, or with having no TERT and all this. So we really nailed it down to TERT being there and seeing that at the same time something goes on with the mTOR pathway. And at this time, I can't remember the authors. There was also some papers showing that there might be a complex with mTOR. So I think one of them was a Japanese author called Kawauchi. I think it was in 2004. It was a quite complicated paper, but at least at this time it came into light that there might be a connection, which was quite still vague. And we only showed that there's a functional connection, not that there is a physical connection in a complex. But it all, if you go down to that, it all makes kind of sense.
Yeah.
And in our last paper, at least one et al., which we published 2021, we tried to go along this. And since we didn't find, so when we activated telomerase with telomerase activators from TA Science, TA65 and GIN 510, we looked to the mitochondria since we were still interested to see whether the mitochondrial oxidative stress is decreased, but we found it only in one of the 2 telomerase activators, TA65, with complex 1 forward and reverse using the right substrate. But we found an improvement also in for example, gait, because we used a Parkinson's disease model, and in balance and stuff. We found it with both. So we thought it might not be the mechanism. So we looked into autophagy, although it was a bit of a pilot only with LC3, B, and also with P62, which is a important protein, and both going down. And so we think there might be something into that. Although I think it needs more research to find the exact mechanism for that.
Yeah, so the idea with this telomerase, so it's this TA65, does it increase the level of the TERT protein, the telomerase protein?
To be honest, since we used mice and we didn't have any really good antibody available, which worked in mice, we only looked for TERT expression, but that increased significantly with both. And we did this in the Parkinson's disease model from Elisée Maslaya. And we did it also in normal mice, which we treated quite at the end of life. So there were 2-year-old mice. And it really improved their balance. I mean, we had a static rod, not a rotor rod, because 2-year-old mice are not able to do this. And that really improved the balance on the static rod of these old TA65 and GRN treated mice substantially. So they could stay on almost as long as young. So there was no statistical difference, and young were 10-month-old mice. So and so then we went on.
So the mice only live a maximum typically of 3 years. So you're looking very
depending on the line,
leaving less than 3 years. So so you started treating the animals when they were 20 months old. They're old. How long?
No, I can't remember. No, rather 24 till 24 months. So exactly 2 years. And then at the end, they were between 27 and 28 months. So that was really just before they were done.
Yeah, but how long was the treatment period?
3 to 4 months every day orally.
3 to 4 months. Okay.
3 to 4 months. No, we did it quite long, but you have to do it every day. So we used like a chow and mixed it in with water where the TA65 was in, because it's not water soluble. So you have to do it with a little bit of DMSO, but we use DMSO for the controls as well. So
do you know if that chemical has made it to the clinic? Is there any?
To be honest, TA65 is a plant extract from Mongolian milkweed or astragalus. No, it's a highly purified. But the other one, this is a chemically designed molecule on the basis of astragaloside.
Okay, so we don't know exactly which chemicals in the TA65 that's causing.
No.
That, it's fascinating. Most drugs though that are prescribed, their original source was a plant or we mentioned rapamycin bacteria.
Yeah, sure. Mm-hmm.
So all these organisms, particularly plants, produce a bewildering number of chemicals, and that's something I've been interested in. Many of them, I think their function is to keep insects or other organisms from eating them.
Yeah, that's true.
But it turns out some of these, and all the chemicals that seem to be good for health, have a very bitter taste.
Yeah. Okay.
If
you have to say, if the medicine doesn't taste bitter, it doesn't help, right?
Yeah. Now, so you found that TERT does something in the mitochondria, maybe increases, reduces oxidative stress, and you had some evidence too that TERT can affect the amount of the main antioxidant in mitochondria called SOD2 or manganese.
Yeah, manganese SOD2. Yeah.
Yeah. And that's we've done a lot of work on that.
Yeah.
SOD2 over the years. And, this is most of the free radicals are produced in the mitochondria, right? When they're burning glucose you got oxygen, to get things to burn, you have to have. But anyway, so you get these oxygen-free radicals, and they can attack DNA, cause DNA damage. If that's too excessive, it can trigger apoptosis or maybe senescence.
Senescence as well,
and then it'll be interesting. Just before I retired, we'd done a flurry of studies with an enzyme called sirtuin-3.
Oh, yeah, sure. Yeah, sirtuins, yeah.
And it's in the mitochondria. It's pretty much only in the mitochondria as far as we know. And in neurons, if you genetically reduce SIRT3 levels, it's bad for them. It makes them vulnerable to disease.
Oh, really? Yeah, yeah.
SIRT3 levels go down in the brain with Alzheimer's.
Oh, okay. I think Ron DePinho also found something with SIRT1 in his recent study.
Yeah.
One of the molecules which he thinks are important for neuroprotection via TERT.
Yeah,
so it also struck me.
We can briefly talk about that. But anyway, it turns out that SIRT3, it's a deacetylase. It removes acetyl groups from lysine, the amino acid lysine in proteins. And just like, you mentioned protein phosphorylation, which can affect the function of a cell. If it's an enzyme, it can increase or decrease the activity.
Yeah, absolutely.
We found that the SIRT3 increases SOD2 activity.
Okay, that's interesting.
So there may be some link to telomerase.
Yes, absolutely.
Maybe we should go out of retirement and go back to the lab.
Yeah, you wish.
But so you mentioned Ron DePinho, and actually it doesn't matter. I can say it. So I reviewed.
Yeah, sure.
I was a reviewer on their Nature Aging paper.
Yeah.
And actually, he'd sent it to, I think, Nature. It doesn't matter anyway.
It's a great paper. I really like it. So it really continues what we have done, right?
But this shows the gap between our work, your work, and
sure.
Because he's been a leader in the field for years. He's mainly a cancer researcher. And
I know. Yeah, he hasn't done so much in brain, doesn't he?
No, he hadn't done really anything in brain. But he did this nice study, and kind of the bottom line, which you were alluding to, he has this evidence that TERT, the telomerase protein, as we'd found, has functions independent of its reverse transcriptase activity.
Absolutely, yeah. Sure.
And then he has this evidence that it can interact with proteins affecting gene transcription. So transcription.
It essentially works itself like a transcription factor, because it binds to the promoters of the beta-catenin pathway, I guess. And it activates BDNF, and I think in your work, and we also came across BDNF, but it's quite a bit tricky because it's not always in the right cells. So I kept out of it. But
we've done a huge amount of work on BDNF over the years. Particularly, it's very important because it's critical for learning and memory.
Absolutely,
it promotes cell survival, stress resistance, things that are good for your brain function. So what you and I are doing now, keeping our neurons active.
Yeah, sure.
Right now, BDNF levels are going up in the neurons in you and I that we're using to do what we're doing now.
Okay, very good.
This. And then also physical exercise actually is very potent.
Yeah.
And physical exercise is good for your brain.
And also as far as I'm aware, there's also another paper came out recently where Chinese scientists, they also found that there's more TERT in mitochondria during exercise.
Oh, really?
Yeah. And and that is connected to the work of the colleagues from Düsseldorf I mentioned, because they have developed mice. I don't know whether you are aware of that, on a TERT knockout background with just either nuclear TERT or mitochondrial TERT. And although they didn't look into brain, they are working on cardiology. But they found amazing stuff. So that was really interesting, because we had also used these shooters.
Yeah,
in
I
cell
missed that.
culture.
That is, so they're working on the cardiac cells. So in a general sense, the cardiac cells are like neurons. They're excitable.
Yeah, and the postmitotic, right?
They're postmitotic. And
Absolutely, yeah.
so we have, skeletal muscle, heart muscle cells, neurons.
Yeah.
And these cells, more than any cells, experience a lot of stress.
Yes, absolutely.
Because
And
every
you
time,
have them lifelong, right?
lifelong stress, a
Yeah,
lot of stress.
absolutely. Yeah.
But
Sure.
actually, that stress is good for them.
Yeah.
That's why
Yeah, like
exercise.
hormesis, you have that behind
It's
your back.
hormesis. Yeah, exactly.
Hormesis, yeah, absolutely. Yeah.
Actually, since I retired, Gabby, I've written 2 books and I'm working on a third.
Oh, wow,
Yeah,
exciting.
the first one was on the science of intermittent fasting.
Okay,
The second one was it's called Sculptor and Destroyer.
okay.
It's about glutamate, the brain's most
Oh, I
important
see. Yeah,
neurotransmitter.
sure. Okay,
And then, but
interesting.
the third book is on hormesis.
Oh, okay, wow.
So stay tuned. That's going to be a while.
Yeah,
I'm just
absolutely.
getting
Yeah,
going on it.
I will check that out, Mark. Yeah.
But anyway, and that's something I think that's interesting to look at with telomerase. So you're saying that there's evidence that exercise, physiological, healthy stress,
Yeah, exactly. Yeah.
may kind of improve or have some of its effects through telomerase, whether
Third, let's say
probably
third. Yeah. Mhm.
non-telomeric even.
Absolutely. Yeah, I think so. Yeah.
Now, let's go back to history. We didn't mention Liz Blackburn and Carol.
Oh, yeah, sure.
And Carol, actually, so she's at Johns Hopkins, where I'm affiliated with them. I'm an adjunct
Oh,
professor
I see.
there.
Okay.
And so I moved to Baltimore in the year 2000. Okay.
Okay.
And she saw some of our work. She actually didn't. She was very skeptical of our work in neurons.
No, and I just remembered her as well, because you remember in Ron's paper, so the shim paper, it's also a lot about VNT. And there is all this work from Steve Artandi's lab, who said that TERT can influence chromatin structure via this BRG and everything. And I think Carol Greider was very much against it, because I remember there was a paper where she showed no, VNT has nothing to do with TERT. So it's a bit controversial, I guess.
This is the dogma, so I don't know if that was one before they had the Nobel Prize, it doesn't matter. But so, this is the way I think that, they discovered, together this telomerase activity, and it's adding its reverse transcriptase and
exactly the classical
and so in their mind, this is what it does, nothing else.
Yeah, that's what my reviewer of my grants always said.
Well, but I had another very similar experience in our career. We did some work with NF kappa B.
Also a very great molecule.
Yeah, and so what happened was NF kappa B levels are high in inflamed tissues.
Sure, it's an inflammatory transcription factor.
Right. And this was the initial finding. So in the minds of the people who initially found David Baltimore and so on, this is a bad thing. Activation of NF kappa B is a bad thing. No, this was this was the thinking back then.
Yeah, but inflammation is also a response to something, right?
Right, but this is the way it was, that this is upregulating the pro-inflammatory cytokines. This is contributing to chronic disease and so on.
It can contribute, yeah.
So again, that was mainly in the immune cells, they're proliferative. And so we started looking at NF kappa B in neurons.
Okay.
And we found it promoted their survival.
All right, okay.
And it took us, we sent a paper to Science. It took 2 years. We revised it, sent it back, the long delay. Then we finally got the paper in PNAS.
Good.
Which is a good journal.
Yeah, absolutely.
Then, 1 month before our paper came out, 3 papers came out in Science from different big shots labs saying NF-kappa B promotes cell survival.
All right. Yeah. And I remember there was also this hype with TERT and NF-kappa B influencing each other. You remember those papers? Yeah. So also a non-canonical function.
Yeah. So I guess the whole point is a lot of things that happen in cells in pathological situations are actually adaptive responses that are trying to keep the cells functioning and alive. And
brain is different in so many matters. So I just wanted to add to this what we discussed before, that we also looked with a PhD student, then Abbas Ishak, into why there is TERT in the brain but no telomerase activity, because in most cells we always say, okay, TERT is the limiting factor to get telomerase activity, because in most cell types there is TERT all over, whether there's TERT or not. But in the brain we found that there is still TERT, but no TERT. So that's why our explanation. So we used a material from very early fetuses. That was downregulated, which is completely different from all other tissues. So it's also, very difficult to get to this into people's minds, because they just repeat what Liz Blackburn or whoever said 10, 20 years ago, right?
Yeah, and then Liz, then after you got the note, she went on this like it was like a telomerase explains everything, every disease. So, so initial findings, like with aging, so in humans, what can you look at? Blood cells, right?
Yeah, or endothelial cells.
You can do tissue biopsy, but in healthy people, so essentially what she did is measure telomere length in white blood cells, lymphocytes, in patients with disease XYZ, whatever, even, psychiatric disorders, Alzheimer.
Yeah, I know. Yeah.
Right. And so, but she's kind of trying to say, oh, this is a cause and effect thing.
Yeah, but nobody knows.
It's just a biomarker of, say, oxidative. It's like oxidative damage.
Yeah.
Or, other things that, are kind of biomarkers of damage to cells.
Yeah.
Yeah. So that's the way I look at it.
Yeah, sure.
Yeah.
Yeah. I mean, I don't know. I mean, she might change her mind. Last time I met her, she said, yeah, mitochondria are interesting, but she never get around to do anything. And then it's good, because something's left for other people to do, so.
Yeah.
I mean.
Yeah, that's okay. Let's see. What else? You talked about autophagy. So you had this kind of indirect, it's kind of a measure of autophagy, the autophagisome that.
Yeah. So maybe some quite known markers. And to be honest, we collaborated with a former postdoc of David Rubinstein with that. So we were lucky to have him along, because again, autophagy, like P53 and NF-kappa B, are so huge areas. You can't just pop there, and it's complex.
Did you use the TA65, or did you?
Yes. So we actually looked into that in the brains of the TA65 and the GRN 510 treated mice. So we treated them for 14 months, because we didn't really know when the symptoms of the Parkinson's disease appears. So we started at 4 months and fed them for 14 months until 18 months, and then we harvested the brains, and then we looked into the different markers. So, and the most striking initial finding was that due to the telomerase activator treatment, the alpha-synuclein, which is the main aggregate in Parkinson's disease, was really, really going down. And TA65, again, was the most effective. And as I said, since we didn't find any explanation via the mitochondrial route, we said, yeah, so why is alpha-synuclein being degraded? And the most striking one, which came into our mind, was, of course, autophagy, right? Yeah. So that's why we looked into these markers. And when you look to the model, which, as I said, was developed by Elisée Meslaya, the alpha-synuclein in these mice only increases at 12 months. So it's really kind of age-related. And at this time, it seems autophagy might decrease, because that's also known. You probably know this better. In the brain, autophagy goes down with age. And that might be that this is when the aggregated proteins, being alpha-synuclein or A-beta or tau, might increase, because the protein degradation machinery is not as effective. I'm utterly convinced that plays a role.
But did you do any genetic manipulations of TERT in that?
No. I mean, to be honest, I had very, very few funding, so that was a big problem in the UK to get any funding. So we were lucky to get this to get this model, because I was actually looking for the triple mutated AD model, which Ron has used, because our previous paper was on human Alzheimer's, and that would have been a logical continuation of that. But then we thought, okay, we take the Parkinson's disease mice, and that was also okay. Yeah. So what we cannot say is where is the mechanistic link between the TERT and the autophagy. As I said, the TOR complex might be a possibility, but we don't know it for sure, because people have shown that only in either lymphocytes or cancer cells. And as you know, not many people look into brain.
Yeah, I think with this TA65 extract too, unless you can't, this chemical mixture could have effects other than increasing telomere.
Yeah, I mean, although they say it has like 80 or 85 or between 80 and 90% of cycloestrogenol, but I mean, our results also showed clear that there are differences. For example, in the rotarod, the TA65 improved very significantly the performance of females, and the other one, the synthetic one, the GRN 510, improved the performance of males. So there was clearly something. Some people say it might be estrogen, because estrogen activates telomeres. We don't know what it is, because in other features, like in the gait test, there was absolutely no difference, but both activators work the same. So yeah, it's still a lot to do there. And I mean, I found this very interesting. What Ron has found, that there is this inactivation of TERT due to A-beta, and I recently reread a paper from Chinese authors which had shown in 2015 that A-beta can decrease telomerase activity by binding to telomeres. Although they, of course, didn't look into brain cells, so they looked into, so they fractionated telomerase into TERT, which didn't show. Yeah, I know. So, but what I want to say is there might be so many different mechanisms. You know?
Yeah. I guess,
but I'm not an AB beta specialist, so hard to say for me what's true and what not.
It might be good to return to the telomeres for a few minutes.
Sure.
So telomeres, there's so you got this repeat TTA, GGG repeats, and they're kind of in this loop, the DNA loops. And then there's these other proteins, telomere-associated proteins of
sheltering,
which there's, it's called the sheltering complex.
Yeah, TF1, TF2, RAP, and POT1.
I had one lab member Pei-Su Zhang, who unfortunately passed away.
Oh, sorry to hear that.
This recently. But she and Miriam Gorospe helped a little too. She's done a lot of work on senescence.
Okay.
Pei-Su found this interesting that there's one called TRF2, telomere repeat binding factor 2.
Yeah.
And so Pei-Su discovered that there's a splice variant that gives rise to a shorter form of TRF2 than the full length TRF2. So that and this TRF2 short interacts with REST. REST is a transcriptional regulator that it inhibits the expression of many neuron-specific genes.
So, I see.
So when you have neural stem cells, they divide, then they get signal, they stop dividing, they start growing axons and dendrites, so they differentiate.
Sure.
And in the stem cells, this REST is keeping various neuron-specific genes. One you worked on, like tau, for example, or
sure.
Whatever. There's actually hundreds of neuron-specific genes. And so she found that TRF2 affects differentiation of cells by interacting with REST in a way that essentially is pulling REST off and preventing it from keeping the cells in the stem cell stage.
Oh, I see.
So that's what a non-telomeric function of TRF2.
No, I met Pei-Su. I'm very sorry to hear. I met her once in Cold Spring Harbor, where we're discussing her poster. I remember.
Yeah, she
wasn't that old, wasn't she?
No, she had a cerebral hemorrhage. She may have had a congenital weakness in a blood vessel in her brain, but she died quickly after a hemorrhagic stroke.
Sure. Okay.
Yeah, that was
very sorry to hear.
Yeah, and she was still working at NIA at the time.
I see.
Okay, I guess my point there is, in the future, as science keeps going like this,
absolutely,
there's going to be a lot more work going on this.
Absolutely. So you still have a lab, Mark?
No.
So you're really retired.
I retired 4 years ago.
Okay.
So in the mornings I do this. So in the mornings I write and do podcasts. So this neuroscience podcast, Brain Ponderings, it's two things. There's no place people like graduate students, even undergraduate, ever can go to find conversations on topics, various topics in neuroscience with two scientists that one's done most of the work in most cases, but in this case both of us have
that's the beauty of it, right?
And then it's also good for me.
Keeps your brain
keep up on
active. And
then
also still do a lot of reviewing and stuff like that to keep in the field.
And then the rest of the day I do other things.
You should enjoy your retirement as well. So where are you located now, Mark? Still in Baltimore?
Right north of Baltimore. We have 2.5 acres.
Oh, wow.
Trees and,
beautiful. That's a good thing.
And
be in a countryside.
The leaves on the trees are very beautiful now.
Ours are almost gone. So it's a lot of wind at the moment. So it's getting late.
You're a little further north.
Autumn. I guess so. But we had really hot summers. So I think that's the hottest summer we ever had in 2023. And September was still very warm with 30 degrees Celsius and stuff like that. So you can feel the climate change, certainly,
Yeah, I'm actually going to include a little section on climate change in my hormesis thing.
Oh, that's actually interesting. Yeah, I
Some
like
kind of
that.
speculation on what's going to
Yeah,
happen with various species.
yeah.
There's going to be, I just heard on the news the other day.
Mm-hmm.
So the state of Maine in the United States, it's north, right? Northeast.
Mm-hmm. Yeah.
It's famous for lobsters.
Oh, yeah, I know.
Uh,
Yeah, sure.
and then
Mm-hmm.
and then Maryland, where I'm at, is famous for a particular type of crab
Yeah, blue
called
crabs,
the
right?
blue crab.
Yeah.
And
I heard about that. Yeah.
so now it turns out with the warming of the ocean,
They all go north.
yeah, there's blue crabs, and apparently
Yeah.
they're carnivorous on the baby lobsters.
Oh, really? Oh, yeah. So it's
So
all
now the
balance
people
is
who earn
gone,
their
right?
living, they
Yeah,
either
sure.
they may have to switch to blue crabs. I don't know.
Yeah, but which is not the worst thing,
Anyway,
but for the for
this
nature,
is
yeah.
like one example of
Yeah, sure.
already,
Yeah,
this is affecting people's livelihood. And
absolutely. Yeah, sure. Yeah.
yeah. So maybe there'll be some role for telomerase in evolution of some species in terms of their survival or not as the
Oh,
climate.
yeah. I mean, I believe that because you mentioned lobsters. I think telomerase is the secret why lobsters grow their whole life.
Yes, you're right. It's true.
And it's even in sponges, these very low, vertebrates,
Yeah.
sponges, they have even TTA-GGG, so you can apply a trap assay on them. It's
No, that's a
fascinating. I
good
really
point.
like that. Yeah.
Now this goes back to the beginning when you were talking about, telomerase, as our tissues are all formed, then telomerase goes down and we stop growing.
Yeah, exactly. Yeah.
And then, but you're right, there are certain species in the lobsters, this was actually studied, right, and published
Yeah.
on, that telomerase stays up and they keep growing,
Oh,
but
yeah.
they don't develop cancers.
Yeah, so they have a different regulation then, or very good tumor suppressors. Yeah, and I mean, if, as I mentioned in the beginning, if you have telomerase but no genomic instability, then yeah, you might get away with it. It's like the naked mole rat. Yeah,
maybe
it's also living very old.
Cancer researchers there should be someone focusing not on cancer cells, but on or whatever, not on species that don't get cancer and why they don't get cancer.
Yeah. No, I mean, I know it's the same with the naked mole rat, and I think they haven't found the secret yet. I know that they have a lot of hyaluronic acid or something, but nothing remarkable with telomeres or telomerase, as far as I'm aware. I might not
so
have.
Gabby, for listeners who don't know about naked mole rat, there's these rats, they don't have any hair and they live underground. And in contrast to other rats who only live like 3 years, these rats live
28,
20, close to 30 years. And they have it's very interesting. There's this, of course, I'm working the aging field, so I could talk forever on this, but there was this idea that there's some relation between your metabolic rate and your lifespan.
Sure. Yeah.
So rats and mice have high metabolic rate. Their body temperature is higher than ours.
Yeah, that's true.
And then animals with lower body temperature, some like fish in the deep ocean, they keep growing too, and they live kind of indefinitely. But yeah, there was this one paper in Nature Science with saying the naked mole rat has high levels of this. It's hyaluronic acid is something that.
Yeah, that's what I mentioned. Yeah,
mostly women probably, but
yeah, we put that on our skin exactly. Yeah,
it's an extracellular matrix molecule that it can kind of keep your skin soft and flexible and.
Now I also know they have barely any DNA damage.
Joints too.
Yeah, I know. Yeah, and I think the naked mole rat have barely any DNA damage even at high age. So I don't know, maybe they have very good repair mechanisms. So yeah, it's very interesting to follow that up, I guess.
Yeah.
So much to do for aging researchers.
Okay, well, I've very much enjoyed talking with you.
Oh, so
Gabby.
did I. Thank
It was good
you.
to meet you in person. I saw
Absolutely.
your papers over the year, and
yeah, I always nominated you as reviewer because I know there are not that many people in the field,
But what I did is I thought about this. I said, okay, I want to have a podcast on kind of overturning dogma, telomerase in the brain. And that's actually my reviewing Ron DePino's paper got me thinking. I said, well, maybe I'll invite Ron. I said, no. He's
not a telomerase person, right?
Yeah, he's late on the scene though.
I know.
So I said, who can I invite that has kind of worked on that? So I knew about
I'm very pleased. Thank you very much. I really enjoyed that. And anytime, if you want to continue on that, I mean, there's a lot of things we hadn't been able to talk about because it's really a nice, exciting area.
Yeah.
So thank you very much.
Okay, Gabby, and have a good holiday season.
Yeah, you too. Take care.
Bye.
Bye. Oops.